Okay, perfect. So today we're going to go over the protein synthesis transcription stage. This is the first stage of protein synthesis. Protein synthesis is all about making the proteins that we need in the cell. Remember, we've been talking about a lot of like enzymes, and we talked about proteins that are in the cell membrane. We need to make all of those. And over the next couple of days, we're going to be talking about the process of it. But first, let's go over some different types of RNA. So we talked about RNA a little bit in the DNA replication. There's actually three types. There's what's called mRNA. mRNA. This is a messenger. A messenger RNA. This is going to act kind of like a little code of the information from DNA that we're going to need. It is a code or a copy of the information. Okay. Next, we have what's called rRNA. It is ribosomal RNA. Ribosomal RNA, together with protein, is going to help to make our ribosomes, which, if you remember back to the organelles that you guys looked at, are these little things that are stuck on the outside of the endoplasmic reticulum, or they may just be floating around in the cytoplasm. This is actually where we're going to be making the proteins. So the rRNA helps to make ribosomes. Make up the ribosomes. And last but not least, we have tRNA, called transfer RNA. Transfer RNA is going to bring amino acids into the ribosome. So remember, our proteins are made up of a big long strand of amino acids. This is actually going to bring them together so that way we can stick them all together and make our actual protein. Three types of RNA. Notice each of them has the lowercase letter at the front. That's going to signify which type of RNA it is. Today we're just going to be working with the mRNA. These two we'll talk about a little bit later as we get into the translation part of it. You guys need another second, or can I erase? Okay. All right. So we have our different types of RNA. That's a big thing because I'll keep talking about the different types as we go in this process. And it's good to know the different functions of each of those. Now, when we're talking about protein synthesis, we have kind of this main idea that we use. This main idea is that we're going from DNA to RNA to proteins. This idea is called the central dogma. Dogma. A central dogma. So a dogma is not necessarily a process. It's more of this idea that we use kind of as a foundation for the process of protein synthesis. So it's this idea that in order to get proteins, we need to start with DNA and then make RNA in order to make the proteins. We can't just skip and go from DNA to proteins. We got to have the middle step. Now, when we're looking at this, there's two arrows here. So this first arrow is one process, and this second arrow is another process. This first process is called transcription, which is what we are doing today. We are going to transcribe or copy the DNA into RNA. The second arrow here from RNA to protein is called translation. This is where we're going to take the information that we just copied, and we're going to go from RNA and translate it from the language of RNA into the language of proteins. We have transcription and translation. Again, we're just going to do the first stage today. Now let me give you guys an analogy so that this makes a little bit more sense as we continue on. Does anybody's mom or grandma or grandpa or somebody in their family have like a family cookbook with all of the recipes in it? Kind of? So my grandma has this family cookbook that her mom gave her, and it's got all of the family recipes in it, and she loves this cookbook. She wants to keep it safe. She keeps it in her cupboard. She doesn't let anybody else use it, but she makes all of these awesome recipes from it. Like she makes cookies and banana bread and cakes and cornbread and all this sorts of delicious stuff. I want those recipes too. I want to be able to make my grandma's banana bread or my grandma's cookies anytime I want. But she's not going to give me the family cookbook right now, because she knows that I'll probably lose it, or it'll get sauce on it, or it'll get cookie batter on it, or something's going to happen to it. So she wants to keep it nice and safe, but I still want the recipe. I still want to be able to make the cookies.
Take a picture of it.
So you could take a picture of it. Or she could write it on a recipe card. She could write it on an index card and then hand me the index card. The other great thing about this is that she could make a ton of those little index cards with all the same recipe on it and give one to me and one to my mom and one to my cousin and one to her best friend and all sorts of stuff.
She does that because in my family, if you want a recipe, you have to write the recipe card.
Yeah. So my grandma tends to write it for me, and then she'll like mail me the recipe card or something like that, or give it to me like when I went up for over Christmas. I got a couple of recipe cards from her, which is awesome. But she also knows that I will probably lose the recipe card at some point, so I will end up asking her for that same recipe card again somewhere down the line, which is okay. She's fine with that. DNA is our big family cookbook. We want to keep it nice and safe. We don't want anything to happen to it. We don't want it to get destroyed. We don't want it to get messed up. We want to keep it nice and safe and secure in the nucleus. RNA is our little copy, our little recipe card, our little index card of that specific area, that specific gene. So she can't fit the entire cookbook on a little recipe card. She's just going to give me that one recipe for that one type of cookie. Proteins then is our cookie or the cake or whatever it is that we're making from the recipe card. So we're not just going to be able to take the family cookbook and make the recipe cards, because then only one person could make it, and my grandma wants to keep that cookbook nice and safe. So she's going to give me a copy, and the best part about it is that me and my cousin can both be making the same type of cookies in different states at the same time, because my cousin lives in Iowa. So we can both be making the same recipe at the same time, and my grandma's family cookbook is still in her cupboard, all nice and safe. Does that make sense?
Yeah.
So today we're just going to talk about going from the family cookbook to the little recipe card. That's the only stage that we're doing right now. As we keep going, probably next week, we will be talking about how we're going from the recipe card to the actual cookies. So I'm going to show you guys the video first. It's going to be a lot of information, like how we did the DNA replication. We watched the video, and it was kind of a lot all at once. And then we'll break it down again.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin.
This is where it
This
starts to really
energy is
speed up.
provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA.
I know.
Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
That was weird. It was like all stopped and it's like it was ah!
I know. Like it starts slowly putting everything kind of on the DNA strand, and then it just kind of zooms away, doesn't it? All right. So that was a lot of information that they just kind of threw at you. So let's start to break it down just a little bit more. Okay? All right. So here is our lovely DNA strand. There we go. Our lovely DNA strand, there's all of these nucleotides in the middle, making kind of this ladder. There we go. Now, the important thing to remember with this is that we're not doing DNA replication. So we're not going to unzip the whole thing and replicate the whole strand of DNA. We don't need to copy down the entire cookbook. We just need to copy that one recipe. So we're just going to focus on this one section called a gene in our DNA. So the rest of it can still be bound up into the double helix. We're just going to kind of straighten out this one little part here so that we can focus on this one area. Okay? When we're doing it, the first thing that it was talking about was what we call a promoter, and it called it the Tata box. So that's like this little area here. And its sequence is TATAAA. So Tata. This is our promoter. So think of it kind of like the primer. This is the area that we're going to start it. Is at the Tata box. Then we had all of these, what they call transcription factor proteins, latch on. And they gave you guys a whole bunch of names. We're not going to go through all the names of each of them. But in general, we call them transcription factors. So I think that they had it as TF2D or TF2A. means transcription factor 2, whatever it is, for the name. Okay? So those are all going to kind of latch on. They're kind of like these big blobs. Those are going to latch on around that Tata box. Transcription factors. Next, we had this really big enzyme that latched on. So we'll draw this enzyme really big here. This is called our RNA polymerase. Do we remember what the DNA polymerase did in DNA replication? Lay down new nucleotides. So what do you think this guy's going to do? Lay down RNA nucleotides. That's his job. Okay. So now, let's see, we've talked about types of RNA, we've watched the video, we've put everything on the strand of DNA. So we've got initiation, we got everything kind of started here. Everything's prepped and ready to go.
So RNA, what is RNA polymerase?
RNA polymerase is going to be the one that's going to add the RNA nucleotides. So that's what's going to be building our RNA strand. I'll let you guys finish taking pictures of this stage here, and then we'll keep working off of this. Wonderful. Okay, so now we've got everything kind of already on the DNA and we're prepped and ready. We got to actually get it going then. So let's see, let's use green. So in order to get this started, they were saying that we have to use ATP, and that's going to kind of link on to both kind of the RNA, but it kind of linked on closer to the DNA too, in order to get this whole process moving. So once we
so where does it like does it go over the part where it overlaps, like the DNA or the RNA polymerase overlaps like the Tata box? Is that what it does?
So this had a really great picture of it. Where did it have it? Okay. So see here we had, I should really draw this underneath then. So we had our transcription factors all on the Tata box. And it kind of led where the RNA polymerase is going to kind of lay down. These guys are going to help the RNA polymerase make sure that it gets in the right spot. Then we're going to have all this ATP kind of come. And if you'll notice,
reduction of ATP into ATP.
It's going to start to link on here. And when ATP kind of attaches here, it's going to kick off one of those phosphates, which gives us the energy. We got a bunch of those ATP molecules that are going to come and they're going to give us a little bit of energy, give it a little bit of boost in order to start the process. So then this guy here, this RNA polymerase, is going to start moving to the right in this case. Did I lose anybody? Now, since we are only looking at such a small area of the DNA, we still need to unzip it, but we don't need this big enzyme helicase in order to unzip the whole thing for us. RNA polymerase is awesome in that it can actually do that all for us. So it'll come, it'll kind of break this bond here. And inside the RNA polymerase, it's going to start to unzip it a little bit. Just a little bit. And if you'll notice, it's all held within the enzyme. So we don't have any area that's unzipped that is outside of the enzyme and could have mutations happening. It's all going to be within the same RNA polymerase. It's going to unzip a little bit of bubble. And then we can get in and we can start adding in our RNA nucleotides. So which direction do we build nucleotides?
5 to 3.
5 to 3. Very good. So if we're building in this direction, we have to be building towards the 3. So what's our template strand?
3 to 5.
Very good. We have 3, and this end must be 5. We can also add in the other side, because you know it's anti-parallel. So we have 5, and we have the 3. Does that make sense? We're adding it in like we did with the DNA replication. Now, as we're doing this, it's actually really cool because as this RNA polymerase is moving, it's unzipping and re-zipping that DNA back together all within the same enzyme. So as this moves down, we don't want any unzipped parts sticking out the end of the RNA polymerase, so it's going to zip it back up and close it back up at the end.
Do both sides get RNA stuff? Does the leading strand get RNA and the lagging strand?
So you're asking if this strand is going to get RNA too?
Yeah.
We don't need to copy that side too, because we know that these are complementary. So we only need to copy one side of it. Good question. Now, as we're building onto this strand, and if we're zipping the ends closed behind us, it's going to kind of start to hang off the end. And you'll notice in the video here, it kind of stuck out the end like a little tail. And the tail started growing. Right here. So it kind of sticks out the end of the RNA polymerase and kind of floats away. Just hangs out in the nucleus. There we go. Does that make sense? Did I lose anybody? This is our RNA strand, and specifically it's mRNA. So the red is our mRNA strands that we're creating. That's our little copy and our little message of the information. We are done with elongation now. I'll let you guys do pictures of it. Awesome. Getting to the end here. We just have one stage left, and that's termination. That's our ending point. So we got to figure out how to get this off of the DNA strand. And in order to do that, we are going to use another enzyme. So in the video, it kind of showed that the RNA polymerase got all the way down the gene, and then it just kind of fell off. It's not just really going to fall off. We got to use an enzyme in order for it to terminate. To push it out?
Yeah.
So here's what happens to get this whole process to end. Ready for it?
Yep, I'm ready.
All right. We have this enzyme, we'll draw it over here, called a 5' exonuclease.
Whoa.
5' exonuclease. Okay? So what this means, nuclease, like nucleotides, and exonuclease means it's going to kind of start to destroy those little ones.
Wait, what does it do?
It's going to destroy the extra nucleotides, starting from the 5' side.
Starting.
starting from the 5' side. It's going to destroy the extra nucleotides starting from the 5' side. So, for this RNA polymerase doesn't know where the end of the gene is. It knows where the start of the gene is because we have that little ta-ta box, but it doesn't know when to stop. Because this RNA polymerase can jump to any other gene that we have in our DNA strand and copy that little area too. This is its job. The 5' exonuclease knows exactly where this specific gene needs to stop. It knows exactly how much we need to copy in order to get the entire recipe and make sure that we have all of the necessary steps and all of the ingredients and everything that we need. So it'll come in and say, this right here is all that we needed for this gene. So maybe it's 100 nucleotides, maybe it's 200 nucleotides, however long that gene is.
And it'll cut it off.
And it'll cut it off right there. So this guy here, the actual gene part of our mRNA is going to kind of float away into the nucleotide or in the nucleus. It's just going to kind of hang out for a little bit while we deal with the rest of this.
Wait, so with the RNA, so it just goes in and like cuts off a little piece of RNA that we need, or does it also go in and stops the RNA?
So the first step is that we're going to go in and we're going to cut this to make sure that we have the chunk that we need, and then that's complete. So this part can float away into the nucleus and hang out there. The next step is that this 5' exonuclease is actually going to come and break apart all of these nucleotides that were just put back together. So it's going to kind of follow the RNA polymerase and actually go into the RNA polymerase in order to destroy all of those, break all of those apart.
Wait, what goes in there?
The 5' exonuclease is going to start breaking all of these little extra nucleotides apart. We could still reuse those little nucleotides later if we wanted to, but it's just going to break them apart so they're not in one long strand. As we do this, once this guy catches up to the RNA polymerase, everything is going to disassociate. So it means it's all going to kind of separate off of the DNA strand. And we have our completed mRNA copy of the information. Okay? So that's the termination process. What questions do you have about this process?
Wait, so if that like wanted to like cut the strand or whatever right there, wouldn't then the rest like floats away?
So this chunk's going to kind of just float away from the DNA strand. It's still going to stay in the nucleus,
Okay.
but it's just going to float away for a little bit.
Okay.
Is it gonna come back?
Come back into the nucleus?
No,
Or-
is it
Like stick back together?
Yeah.
No, it's not going to stick back together. We have our completed copy of our information.
Okay.
Okay? All right. Any other questions about transcription?
Looks good.
Wonderful. I will finish my little